afternoon session of our long COVID. Welcome back to the late afternoon session of our long COVID consortium. Uh, this morning we had physicians and patients in to talk, and now we're going to. Now we have a group of innovators and researchers, and we're going to talk about the path to a cure, to treatments. One of the things, for those of you who weren't here this morning, we talked about the need to do something different at this agency, which is not just to develop science out of NIH, but actually to go out and talk to physicians, talk to patients, and find out what's working on the front line. And we had a very successful meeting just now. And now we have a new group of people, and why don't we start over here? Why doesn't each person introduce themselves and talk about some of the things that they're working on? And I do want to say we have Senator Todd Young with us today. During my my confirmation hearings, he asked me to include ARPA-H and discussions about long COVID and bring them in. And we have Dr. Jason Roos, who is from our ARPA-H. So that is, now we're even. Thank you.
Thank you.
Thank you.
Thank you, Mr. Secretary, for inviting us. I'm Hossein Estiri. I'm an associate professor of medicine at Mass General Hospital. I would like to start by stating that I'm here as an individual, not representing my institution. I'm here as a scientist, deeply rooted in rigorous scientific evidence. I'm an empirical scientist. I believe in gold standard science. And I'm also have personal experiences with long COVID. I'm not a clinician. I'm a data scientist, social scientist, but I work very closely with a lot of clinicians. I have all of my perspectives are basically from my research over the past 5 years on long COVID and deep appreciations of the complexities of human health. Should we go a little bit about what we do? All right. So most of my work is on defining the problem, which is I think is part of the bigger problem of why all of the investment hasn't resulted or anything. I believe that what we do is that we use computational basically techniques and AI and big data sets to define complex temporal human phenotypes. And I believe one of the main challenges is that we've been trying to target something that is elusive, has never been decided, they're defined, and it's all over. So I think that a part of lack of success is it probably is one of the hardest thing to actually ask for treatment when you don't still know what is exactly out there, because it manifests in several different ways, and it can be very individual. Treatments can be very individual also. So we have to, we probably are talking about a lot of different subphenotype definitions here, and we've never had enough, power in our data in the legacy approaches to define this disease so far.
Thank you very much, Doctor. And that was one of the themes that we hit on this morning, is that this is a constellation of symptoms that each patient needs to be treated individually, and that, we don't have biomarkers and we don't have, many of the characterizations that we have for other diseases. So thank you for adding to that. Now, Bruce Patterson, who's also a clinician who has had great success in treating COVID patients from all across the country.
Thank you, Secretary Kennedy. It's my pleasure to be here. I am a physician scientist. I was trained at Northwestern in viral pathology. I went in to start my research career in HIV in 1990. So I've lived through these pandemics, and the words you said in the previous session couldn't be more true. The fact that we're sitting here together discussing the issues and the struggles is such an enormous step and so different than how HIV was treated back in the day. I went on to do a head of virology at Stanford, where I continued my HIV research on reservoirs, viral replication, and what I call replication competence, which is the ability for viruses to make more of themselves, to replicate. And at the time, we started studying monocytes, which are a white blood cell, and harbored many viruses, including what we published in 2009 with hepatitis C of all viruses. And it brought an issue to me was that these monocytes, which are human scavengers, basically can take up or get infected with viruses. And so when I got back from China in January 2020, we used 158 biomarkers to immunophenotype acute COVID. And we found that patients at 60 days and 90 days may have been better, but by no stretch of the imagination was their immune system normal. It was highly dysregulated, as Dr. Redfield had mentioned, and it was abnormal in a very different way in acute COVID. So we took these 500 patients at a time where there was only one variant, and we modeled using machine learning and AI the two algorithms that set apart acute COVID and this population of individuals who had symptoms at 90 days, now recognized as long COVID. So even then, we had the two algorithms that were a composite of cytokines and chemokines that were biomarkers that allowed us to separate long COVID. We then went on to model chronic Lyme, chronic fatigue syndrome, and now we're working on mold mycotoxins, as was mentioned in the previous meeting, where we can actually use the diagnostic to obtain a pure population of patients with long COVID, chronic Lyme, and other chronic inflammatory conditions. That is paramount to doing, obviously, very informed clinical trials where patients get enrolled properly. We also use that data in now 20,000 patients. We've treated 12,000 patients. We have a database of 20,000 patients where we used that information in a data analytics portal to develop a therapeutic that could address long COVID. And we're very excited to be on the precipice of launching our FDA trials in that combination drug that was approved to proceed 18 months ago. And it's, it's fortunate that we're sitting here because this trial could have been done, but I have spent the last 18 months trying to raise money to fund this trial. But long and short is we discovered a new mechanism through our data analytics that there can be viral persistence in the absence of replication. So there's fragments of virus, viral RNA in monocytes. There's fragments of viral protein in monocytes. We showed that in long COVID. We showed that in post-vaccination syndrome. So that's almost proof that you don't need replicating virus to basically have the same symptoms as long COVID. And the clincher was we just found the same mechanism in Lyme disease. In other words, retention of Borrelia protein in long-lived non-classical monocytes that their sole reason to live is to traverse to the blood vessels and cause vascular inflammation. And that's how we're treating long COVID and chronic Lyme. But it was very prophetic that you, Mr. Secretary, brought together ideas about long COVID and Lyme because personally, I think they're very much related from a pathogenesis standpoint.
And what is the name of your drug?
It's a combination of Maraviroc and torvastatin. And so it was similar to what Dr. Redfield was talking about, but the base is Maraviroc, a CCR5 antagonist that has a very unique role. It was used as an HIV entry inhibitor, but it's a profound immune modulator, and it prevents inflammatory cells from migrating all over the body. Most importantly, it reprograms monocytes away from a pro-inflammatory phenotype, reducing this immune dysregulation in long COVID patients. And last, the lymphopenia, or the low levels of CD8 T cells that you see in long COVID, Dr. Iwasaki has published that as well, is a function of the CCR5 pathway. And that lymphopenia, low lymphocytes, gets restored by using a CCR5 antagonist like Maraviroc.
Thank you, Doctor.
Hi, everybody. I'm really grateful for the opportunity to be here. My name is Michael Peluso, and I'm a long COVID physician, researcher, and clinical trialist at the University of California, San Francisco. I've overseen our long COVID program since way back in March of 2020. And our program is really focused on, two things. First, understanding the biological root causes of long COVID. We also have evidence for things like viral persistence and ongoing immune dysfunction, immune dysregulation. And secondly, conducting clinical trials with the goal of getting people better. We are involved with about 7 different clinical trials, really trying to push the envelope on long COVID therapeutics. And, I'm really here today to advocate for what's been mentioned already, which is the importance of really scaling up our portfolio of clinical trials for people with long COVID. So I think that there are basically three things that we need to do that. The first is really to implement a flagship biomarker program to figure out who is most likely to benefit from each of the types of clinical trials that we might do, and to also get diagnostics into the clinical setting so people can get a diagnosis and we don't continue gaslighting our patients and telling them that they're, this is all in their head. The second is that we have so many leads on what actually needs to go forward into clinical trials. Dr. Bhattacharya mentioned this before, and we need to be chasing them all down right now. The pace of clinical trials has been too slow. We need to get more trials into the field like this year. To do that, I really think we need to invest in a bottom-up approach to clinical trial implementation, with a variety of different groups that are each running their own trials, but with shared enrollment criteria and outcome measures so that they can be directly compared when we have results. And then the third thing that we really need to do, which also came up this morning, is to promote industry investment and engagement in this problem. We saw for HIV how important it was in the 90s to have pharmaceutical partners on board with developing drugs and investing tremendously and getting people onto ART, figuring out what treatments would work. We need that level of commitment for long COVID. Thank you.
Good afternoon, everyone. Sorry.
Go ahead.
Go ahead.
I know, I'm just introducing you.
Well, good afternoon. I'm Jason Roos. I'm the acting director at the Advanced Research Projects Agency for Health. First, I want to thank everybody for taking the time to come here. The patients, the providers, in the previous session, all of our research colleagues here, and Senator Young, appreciate your support as well. ARPA-H was born in the model of DARPA with the goal of going after the hardest problems in the healthcare space. And our mandate is to really focus our investments on those high-risk, high-impact research areas that can really push the boundaries beyond those incremental improvements and really be transformative. And I can't think of any other problem right now we're dealing with in long COVID in terms of what fits into that bucket. But we are not in any way, shape, or form going to solve this problem on our own. My whole of government colleagues here, Jay and Marty and CDC and others, this is a problem we all have to tackle together. But importantly, as was brought out earlier in the earlier session, it's going to be very important to involve you all and public-private partnership, engaging with the community outside of the government as we try to tackle these problems. So I'm here today to listen to what you all think where we should be placing our investment. How do we maximize the use of our resources? Who do we need to be partnering with to really try to make some significant progress here? And I can say that we are committed to really trying to move the needle and being true to our model. And the output from today is going to help inform the investments that we're intending to make in the very near term on the long COVID problem. We were talking earlier there, we're already starting to look at this idea of repurposing, which came up before. We have some investments there on the rare disease space. How might that be applied in this world? And then certainly on the biomarker front, that's an area of great opportunity. So again, I just want to thank you all, and thank you, Mr. Secretary, for your strong interest and push in this area.
I'm Jay Bhattacharya, I'm the Director of the National Institute of Health. I already gave comments earlier, so I don't know how much I should repeat myself, but I'll say that the need of patients that I've heard from on this is just tremendous. Like it's the heart and soul of everything that we at the NIH are working on this. Bobby, your passion for this is I mean, truly inspiring. I think the key thing in my mind is that there's a lot to learn from patients, a lot to learn from from clinicians who manage patients, the Recover TLC program, the NIH's program on clinical trials and long COVID. We did essentially a listening session where we got literally hundreds of possible agents, drug agents, a whole host of other therapies. There's a lot of expertise out there. And Dr. Peluso, you mentioned the imperative that we start evaluating them rapidly. That is absolutely true, and we've been way too slow. Despite spending billion and $1.7 billion, we've been way too slow at evaluating them. And I think let me just make a defense of evaluating them, just because I think it's important. I think it's very important to listen to patients. And I think it's very important to get the therapeutics out there as rapidly as we can. But if we don't evaluate them, things that don't work will be given to patients as false hope. And so so the idea is for us to work together with the patient community, to work together with clinicians on the front line, but we have to work much more rapidly in academics than we're used to do this right. I've been listening. I have tremendous hope. There's so many ideas that seem to me like they're very, very promising expertise that's been developed over the course of now years. I think we will have an answer, but it'll take way too long if we move at the normal pace of science. The kinds of consortia that have been developed, the sort of clinical trial network, the capacity to use these networks to accumulate our learning and then communicate with others. That's what I'm absolutely focused on, and I'm delighted that Secretary Kennedy has convened these panels just so we can continue to have that engagement. The website you were talking about is going to be fantastic, I think, for patients to get to see what's available, and we're just going to keep moving forward. For all the long COVID patients out there, we're going to have an answer for you.
Well, Mr. Secretary, thank you for your leadership on this and for not just keeping your promise, but going above and beyond the call of duty and highlighting the importance of this issue. We know that millions and millions of Americans have been adversely impacted by the after effects of COVID, and I do think it's going to take an all of government's multi-stakeholder engagement to address this unique challenge. NIH, ARPA-H, our university research community, private sector, to have them all here and figuring out ways in a very transparent setting to cross-cut this issue, to tackle these challenges, is going to be essential. So I wanted to deliver my word of gratitude, Mr. Secretary, but also assure you that members of Congress are engaged in a bipartisan way on this issue, very interested in the development of therapeutics and other ways we can tackle this, prepared to resource evidence-based interventions where we can, and really eager to engage with our constituents, whether they be members of our family or the business community or rank and file Americans who just are at wit's end trying to make life work at the time that they've been debilitated by this condition. So thank you. Sign me and other members of Congress up to continue this positive progress.
Thank you, Senator.
I'm Jim O'Neill. I'm the Deputy Secretary of HHS and Acting Director of CDC. We've learned a lot about symptoms of long COVID, a long list, an informal list so far. And I would like to add one symptom of long COVID that I don't think we've mentioned yet, and that's alienation. So many people are suffering and people around them don't believe them, don't take them seriously. It adds alienation to all of their other symptoms. And I want to thank you, Mr. Secretary, for convening this conversation, putting it on the web. I'm hoping you'll provide validation and support for people who are suffering from alienation, and also hope and optimism that we are putting more facts on the table, bringing more expertise, taking unstructured data and starting to add structure to it so that it can lead to more solutions that everyone will have access to. And you've asked us to build a website at CDC to put all this information together, information from patients, from providers, from manufacturers, about repurposed drugs, about symptoms, about possible causes. I think this will be a great aid to all of our research. We have our Chief Technology Officer, Zach Terrell, in the room, who's listening very carefully. We have Kristen Honey, our Chief Data Officer. We are taking all this information and suggestions on how this new website can best be a central repository for the world to gather this information. So thank you all for participating in this.
Just want to echo, thank you everyone for coming. My name is Ken Callahan. I work on policy for Secretary Kennedy. As he had mentioned, the first session was very much focused on giving providers and patients a voice and a platform to share what's worked. And this session is very focused on finding a cure, improving diagnostic testing, and just seeing what we can be doing on the government side to improve innovation in this space. So just want to echo the Secretary and Deputy Secretary, and thank you everyone for being here.
Good afternoon. I'm Darcy Johnston. I'm the Principal Deputy Director of the Intergovernmental and External Affairs Office. Thank you for being here today.
Thank you, Mr. Secretary, for the opportunity to be here and for convening these two panels on long COVID. So I think we can all agree that long COVID is a serious national crisis. There are at least 20 million Americans who are suffering from long COVID. Some are actually disabled because of long COVID. Long COVID affects people across political lines and in every corner of the United States. Wherever I go, there's always people with long COVID. While the NIH's Recovery Initiative was a well-intentioned start, it was very clear that its results so far have not matched the scale of the investment and the urgency of the crisis. We think we can all agree on that. The number of people suffering from long COVID actually continues to rise. It's a misnomer, or people don't really understand this, that even in 2025, people are still getting long COVID from COVID-19, even in 2025. It didn't go away. The number of people who are suffering from long COVID actually continues to rise. And these people need answers yesterday, need answers and treatment yesterday. To that end, I have like 3 suggestions or initiatives for your consideration. First, facilitate data liberation. And what do I mean by that? Create a national data commons for long COVID. A major roadblock to progress on long COVID is that the Recover's vast data set is literally locked for only Recover investigators. And that data was paid for by US taxpayers. I paid for that data. All US scientists should be able to access that data to help us solve the puzzle of long COVID. 2, fund a diverse portfolio of drug repurposing programs. This is really, really important. We've made a lot of progress on AI over the past several years. I don't see that leveraged to really help us solve the problem of long COVID. We can and should do it. It's not beyond us. It's not beyond the might of the United States to solve the problem of long COVID. It's not beyond us to solve it. And we can and must solve it. And how you can do that? You can really study using AI and existing data, thousands of drugs for repurposing, identify the most promising candidates, and then give them to NIH and say, instead of like testing 100, which you will not have the resource to test 100, 4 is not enough, but maybe these are the top 15 that can really, you can put your money in and then try to investigate those. 3, really galvanize research on studying the epidemiology and mechanisms. While we focus a lot on treatment, and that's really, really important because people are hurting and they want treatment yesterday. They really want treatment yesterday. But there is really, we need also to understand epidemiology and mechanisms. For example, somebody said, oh, like a lot of people in my family have long COVID. Why some people get afflicted with long COVID and most of us are just fine? I got COVID, I'm fine. Most of us, when we get COVID, we're fine. Why do some people get long COVID and some people don't? We need to understand that. We also need to understand what happens to people 5 years down the road after long COVID, 7 years down the road, 10 years down the road. These are really, really important questions that need to be solved. We are in a race against time. We must make progress on long COVID. With board leadership, I'm grateful to you and to you, Senator, and to the rest, to the NIH and to the rest of you for actually convening this. Enormous gratitude for doing this. With your leadership, I believe we can move the ball forward on long COVID. It's not beyond the power of the people in the room and the United States people to really solve this problem. We can and must solve it. Thank you.
Will you just talk about the database that you're talking about that we don't have access to?
Correct. So Recover has in the observational arm and also in recruiting patients has amassed troves of data, but those are primarily available to only Recover investigators, and they make really only the secondary or tertiary data sets available to people outside of Recover. If we are really truly serious about solving the puzzle of long COVID, you would want the best and brightest minds in the United States to really get access to that data and help you solve the puzzle of long COVID. Of course, no shorting privacy and security, and must be done correctly and must be done by upholding the high standard of security and privacy. We're not cutting corners, but US people who are actually invested in helping you solve long COVID should be able to access that data and help you unlock or help you solve the puzzle of long COVID. That currently is not the case.
So Dr. Ali, yeah. So
What is the impediment?
There's
That seems like a no-brainer.
yeah, no, this is what the real-world data platform that we're working on is supposed to exactly be for. Not just long COVID, the recovery data, but just the data sets that the United States government has for on a whole host of a whole panoply of data sets that we have, making them available to researchers. It's not just for long COVID, for chronic disease after chronic disease. We need to unleash those data resources so that researchers of the world can use them. I mean, I can tell you, before I was an NIH director, on the outside, I was quite frustrated. And I can hear the same frustration in your voice. I'm absolutely committed to making that happen. What you said also before about the patient privacy is very, very, very important, right? We made essentially a compact with patients that we're not going to violate their privacy. But there are ways to do that now.
Yes.
And make those data available, protect the privacy, and then allow researchers to. That's what the real world data platform they're working on is exactly about.
May I add a comment to that data because I've worked with different networks as repository. I think that one of the main challenges is validation. The data, until it's not connected to the patient, and you cannot go back and trace it back and validate and potentially follow up the recovery for the patient, it's not useful. It's not a centralized solution, in my opinion, is not useful. It has to be a federated way so it can go back. Look at the entry C, 4C, all those practices. I don't think the answer is in secondary use of data because a lot of unvalidated work was out during the pandemic, and a lot of misinformation, honestly, went out through even scientific publications without any validation. And I really want to emphasize that it's important because this secondary data, even the surveys data, these are not, the surveys are they have their own biases, but secondary data, EHR data, claims data, they were not collected for research. There's a lot of inconsistency. There is a lot of quality issues. I just wanted to say that there are solutions. I completely agree that we should utilize the data, but I don't think it's federated way is going to be traceable back to the patient to validate.
We can discuss that offline, but I think that those secondary data actually could be useful if they're analyzed properly and within ways that are. We can talk about that. These are the kind of problems that we're tackling in this real-world data platform.
Mr. Secretary,
Yes.
do you mind if I ask sort of a
No,
question of the group?
You and I are co-chairing this.
Oh,
so-
very all right, kind of you. So thank you for hosting again. This is thematically, I think, a good point to ask a question, not only how we can repurpose existing pharmaceuticals, but whether there are ways to get more companies at the table to help solve these problems. It seems to me that the FDA does amazing work. There's been some, I think, thoughtful reflection about how to reform the FDA or how to impose leadership to cut through bureaucratic delays and bring more solutions to those who need it. Mr. Secretary or others, are there things that we can be doing, perhaps a pharma roundtable at some point to bring stakeholders to the table and figure out collectively how we can accelerate U.S. long COVID research through FDA reforms?
Yeah, I mean, this is exactly the question that I asked at the last session: is how do you bring in a public-private partnership? How do we incentivize private companies to get involved, and particularly when there's repurposed medicines involved, where the patents may have expired or somebody doesn't own the patent? It's a little trickier than it was during COVID, when we were just focused on vaccines, and there was a huge profit potential. But maybe other people have ideas about how to do that.
So yeah, existing drugs, but also drugs in development. Are there things we can do through the FDA process? Maybe others have thoughts about that.
I think, you know.
Do you mind if I
oh.
introduce myself?
Yes.
Go ahead.
I think we still have 2 more people. So I'm Akiko Iwasaki, professor of immunobiology at Yale University School of Medicine. And I'm also director of the Yale Center for Infection and Immunity. We started the center 2 years ago to tackle chronic diseases that happen after an acute infection like SARS-CoV-2. We also realized that many other pathogens can cause similar outcomes, which we're calling post-acute infection syndromes, from Lyme disease, Epstein-Barr virus, and multiple other causes. What we're understanding from long COVID research is very precious because it informs immune dysregulation for other types of post-acute infection syndromes. What is happening, seems to be, is that there is a fundamental inability in some people to clear the virus at the acute phase, which then leads to persistence of the virus or the viral antigen or the RNA, that then leads to things like herpes virus reactivation, Epstein-Barr virus, as well as induction of autoimmunity, autoantibodies that we can detect in about 20 to 25% of people, and also chronic inflammation that's not resolved. So we're seeing these features from multiple studies, and we believe that tackling these root causes of disease is going to make the biggest impact in patients' lives, because it's not just a symptomatic treatment, but getting at the root cause and getting rid of the cause of disease. So for instance, for viral persistence, we can deploy things like antivirals, monoclonal antibodies. We also heard about Invivyd, which makes Pemgarda, and they have a next generation monoclonal that's even more potent that we can trial in patients with long COVID. And for people who unfortunately already developed autoimmune diseases, there are many, many amazing drugs in the space of autoimmunity that we can retool and repurpose and to do a randomized clinical trial. And for herpes virus reactivation, we really need to develop better drugs and vaccines against these herpes viruses. For instance, investment into antivirals is key for preventing future pandemics and future occurrence of these post-acute infection syndrome, as well as developing better innate immune agonist, which allows your host cells to remove these pathogens on their own. Again, also using immunotherapeutics to remove the persistent virus, and whether it be SARS-CoV-2 or Epstein-Barr virus. We also, I believe that more support in basic research is needed. So from the NIH, we need to really look at root causes in a more rigorous fashion with much larger cohort of studies, in addition to doing randomized clinical trials and trying to do deep immune phenotyping so that we can identify people who benefit from a particular medication and who don't. So we won't be wasting people's resources and time and energy into giving a wrong treatment for patients. Just as an example, if somebody has an autoimmunity you don't want to be giving that person antivirals. It may not be relevant, and vice versa. So understanding what they have and what the root causes are important, and biomarkers, many of us already talked about this, but having a validated biomarker to identify people who have persistent virus antigen versus people who have autoantibody versus people who have chronic inflammation will go a really long way to doing a targeted randomized clinical trial. And for engaging industry, academia, and regulatory agencies all together to expedite this process, because as many of us already said, we really needed this randomized clinical trial yesterday. So I agree with it. And I'm here to serve and be a resource for this group.
Jay, do you want to respond to that?
I mean, I agree with all of that. I've learned a lot from your work over the years, and I'm grateful for it. So thank you. I think we have to invest in all of the above. It really is a tragedy that we've spent so much money and time and not gotten those validated biomarkers. But I don't want to wait. I don't want to wait. I want those randomized trials happening now as fast as we can. We can't do hundreds, but we can do a lot. I've heard like the possibility of private collaborations. I think that's one way to accelerate things. We have to do all of the above.
Dr. Peluso, you had a comment about how to bring in a private.
Yeah, I just want to make a quick comment. So, I spend a lot of time as a clinical trialist doing kind of proof of concept or early phase 2 trials, knocking on doors of different companies that have drugs that I think can and should be tested for this condition. And I perceive this hesitancy because this is considered a really risky investment on the part of a lot of these potential industry partners. The question that comes up by far the most often is, how do you know that the people that you enroll in this trial actually stand to benefit? And that sort of gets at this biomarker issue. I think that they are concerned that, a bad outcome in a long COVID trial, which is not going to be straightforward, could harm their product development pipeline. So I really like the idea of actually having a roundtable event like this with industry partners to really get a sense of what their concerns are and what would facilitate this. The last thing I'll say is it's not at all clear what the FDA would accept as an outcome in a clinical trial for an indication. And so guidance from the FDA, perhaps as part of a roundtable like that, would be extremely helpful, I think, in engaging that key group of stakeholders.
Dr. Patterson.
I think that was such a great idea. I think in terms of biomarkers and in terms of how we use our diagnostics, it's twofold. Number one, you need a diagnostic to stratify, ensure that you're enrolling long COVID patients into a trial and you're not enrolling Lyme patients into your long COVID trial. Okay, that's been the focus of our trial design. Number two, you need a biomarker or a collection of biomarkers, as in our long hauler index, that shows response to therapy. So you take it from, you have a companion diagnostic in addition to a diagnostic that can stratify. So it goes much beyond discussion. Maybe it's because we're kind of beyond what biomarkers should we use, where we're actually deciding how to deploy that diagnostic, deploy those biomarkers. And like I said, one to stratify, get a pure long COVID population, and one to monitor response to therapy. And that brings up the point. I think long COVID and the term PASC are two different definitions. I think SARS-CoV-2 in of itself is capable of a post-infectious condition, Dr. Iwasaki mentioned. I think PASC encompasses the fact that SARS-CoV-2 causes profound immunosuppression. CD8 T cell counts in the single digits when we started in February of 2020. And of course, you get the herpes family virus reactivation. You get Lyme recrudescence or reactivation. And now mold mycotoxins we're studying. So that is truly post-acute sequelae of COVID. That's PASC. I think with long COVID, it's pure COVID. It's not Epstein-Barr reactivation. It's not CMV reactivation. It's not Lyme. And I think that's how we've kind of framed our development of diagnostic to decide on that. So we get pure long COVID in our trial.
And I would ask all of you, including Dr. Iwasaki, what is your confidence that you can distinguish between Lyme and long COVID and short COVID, for example?
Well, we've published that already in 2 papers, so we're very confident. We've studied over 400 patients in each of those papers, and we have real-world data on over 12,000 patients. So we have supreme confidence. If we had more bandwidth and more people, we would be submitting our drug trial and our diagnostic to the agency at the same time. Right now, we're just trying to get our drug trial started. So, but we're going to use the clinical data derived from our clinical trial for the drug as our clinical portion of our biomarker studies, because our biomarker is incorporated into our clinical trial design.
And can you comment on that, Dr. Iwasaki, because you treat both Lyme and long COVID?
Yes, so I'm a basic scientist, so I collaborate with clinicians who treat both long COVID and post-Lyme. And in terms of diagnosis for post-COVID, it's, it's still challenging. I wish there was a blood biomarker that says, okay, you have long COVID versus you have long Lyme, you have long EVB. But these things are fairly overlapping. So some people may have all of these things going at the same time, while others may only have one of these things. So I think we're not there yet, and maybe your diagnostics works well to dissociate these things, but often patients come with multiple of these pathogens. And the other thing is that people who have Lyme tend to be more susceptible to getting long COVID and other post-acute infection syndromes. So it's a little bit difficult to disentangle all this, but the root cause driver may be shared. For instance, persistent pathogen, whether it be Lyme bacteria or SARS-CoV-2 virus or EBV, if we can treat that root cause, it may still benefit these patients. We may have to use multiple antimicrobial to get rid of all this.
Mr. Secretary, I just wanted to pull on a thread here. Dr. Peluso and Dr. Patterson both indicated they thought it would be helpful to convene a pharma roundtable. You and I can perhaps discuss that offline or work with your team on whether that makes sense. I can tell you I have an interest, regardless of perhaps convening such a roundtable on Capitol Hill. So know that for all watching this, know that that strikes me as something that is worthy of doing.
Dr. Willie Chirpin, do you want to introduce yourself and give us any comments?
Sure, I'm Willie Chirpin. I work for the Secretary and the Deputy Secretary as a science advisor. I actually used to work in Senator Todd Young's office, where he was a champion of oversight on long COVID efforts. And I think the fact that this roundtable brings together the leadership from all parts of HHS shows that this HHS is really committed to solving hard problems, because everybody here, I think, probably agrees that long COVID, chronic Lyme, these are among the trickiest medical problems out there. And we're committed to doing it transparently. So I'm learning a lot from everybody, and feel free to follow up with me after on the concrete ideas that you've all come up with so far.
Dr. Patterson, I know Senator Young has been open about his own struggles with, and I'd love to hear what your experience is and what your experience with your patients is.
I'm running a pharmaceutical company myself, and people ask me, why do you still see patients? I can't tell you how valuable it's been over the last 5 years to see over 12,000 patients, sit in front of them, hear their stories of disability, how it affects their families. There is no doubt that this is something that is so pervasive in this country, and it goes well beyond the 20 to 30 million people who have it, and there's no doubt that it's more than $1 trillion cost to the healthcare, group. So, but the fact is, hearing their story, symptoms, and then seeing their biomarker data, and relating the two, and testing our basic science hypotheses and approaches in a real world case where we're seeing patients who are using repurposed drugs, and we think it's been very successful to date, and we look forward to our trial. But that interconnection between the patient and the science is invaluable, and it's all captured in our data analytics portal, where we're doing a lot of the things that have been suggested here by ourselves. We have our data scientists who are digging into our 12,000 patients' laboratory symptom data and looking at the connections and response to therapy. I mean, subjective responses to therapy are so difficult. This population has been so sick that it's really hard to say, well, I feel better. Maybe I'm better. I don't know if I'm better, but I'm better. But to see their immune system resetting and getting back to normal on our panel is so reassuring, not only to us, but to them that, hey, you know what we're trying right now is at least working and getting your immune system back to normal. And for the most part, we see symptoms following. That, I wouldn't trade that feeling for anything in the world.
And how would you describe your success rate?
People always ask that. And I think early on, we didn't even have a diagnostic that could separate long COVID from Lyme. Early on, I would say, 70 to 80%. It could be even higher now that we're able to say, well, no, you have a long hauler index over 6, or you have high interleukin 8 plus high interferon gamma, or high interleukin 13 plus high interferon gamma, you have Lyme. And being able to make that distinction in our real world data, I think has been invaluable, and maybe our success rate is over 80, 85%. But I think in terms of the clinical trial, that is absolutely critical. So please.
Yeah, no, I think my perspective on this is that it's so important to learn from, the experiences of patients who are trying different therapeutics, and then to, like you said, get those therapeutics into clinical trials that can be done safely and with rigor to prove that a therapeutic does or does not truly work. I think we've seen some things advance to clinical trials based on that. For example, there have been several trials of short-term antivirals based on patient experiences. But the other thing that I just want to kind of state for the group is that, often clinical trials are negative, but a negative clinical trial is not useless. We actually learn a tremendous amount from trying and not seeing an effect because we can understand whether the drug did or did not have a biological effect in the first place. And then we can really try to hone in on whether there's a subset of people, and I'd love to hear Dr. Iwasaki's thoughts on this, whether there's a subset of people that do truly benefit. How do we understand who those people are? And then how do we identify those people for the next clinical trial, which would really give us the best opportunity to find something that truly works for a subset of people? Because as we've mentioned repeatedly in this is a heterogeneous condition. That is a challenge, but that should not be an unsolvable problem. And I think that we can learn even from, clinical trials that don't hit a home run for how to inform how to move forward.
Yeah, thank you very much, Dr. Peluso. So one of those, an example of this is the Paxil-C trial. We did a Paxlovid 15-day course of treatment in patients with long COVID in collaboration with Pfizer. And we did not see an effect in terms of the primary outcome of improving the health of these people sufficiently. However, we did a deep dive immune phenotyping in these people, and we are seeing some signatures that may be predictive of who recovers versus who don't. Again, it's not in everyone who took the drug, but these signatures are then going to inform us what might be the next target that we should go after. And so Dr. Peluso is absolutely right. A failed clinical trial is a learning experience. And we have to learn from each of these because they're very expensive, laborious, and the patient's time and effort, we cannot waste that.
Let me just a brief comment about, because everybody agrees that we need to accelerate clinical trials. We also agree that it's a heterogeneous problem. Paxlovid trial, how long did it take for you to start and do it? Several months, right?
Yeah, so our Paxlovid trial was relatively quick and cheap because we did it in a decentralized trial, which is really like amazing effort by Dr. Harlan Krumholtz at Yale, who specializes in these next generation trial platforms. And this is what I would recommend to everyone, because patients with long COVID and other post-acute infection syndromes are quite ill. And even if they wanted to participate in a trial, they're unable to come to these sites. So we sent the phlebotomists to their homes. We send all the surveys electronically, and the drugs are sent to their homes, remotely. So none of the patients had to come to any given sites to do the trial. And I think it's cheaper and faster. And this is something that we should consider going forward, especially when we're having this discussion with the pharma partners.
Now, my comment is that we can actually do these kind of trials on multiple hypotheses using data. In fact, we have done that, and we found that Paxlovid doesn't have any effect. And we have curated this cohort of the largest curated precision cohort in the country, 150,000 patients. 21,000 of them have unexplained chronic conditions post-COVID that associate with COVID. That's about more than 14%. So we have this data of it's about 80% accuracy, which is still great given the validation. We are testing hypotheses that takes several months to just test in standard ways. I think with the power of data, with the data that is available in all academic medical centers, we can do things like FIWAS type analysis and look at a lot of different things and accelerate the beginning part of the trials and have the hypothesis be driven by data, I think.
This is a high-risk, high-reward public health condition. ARPA-H has a high-risk, high-reward model. This is why you were established, as I understand it. So, Dr. Roos, I wonder whether you have a timeline for initial planning to assist with this public health condition, and whether you might be able to give us a sense of when you might potentially launch your long COVID work.
So we've already begun this discussion in coordination with the Secretary's office and his counselors and the community of interest here. We felt that this forum, and as we were engaging with the Secretary, this forum was vital to help informing that. We don't want to just jump out and presume that we have the best path forward. So I'm very confident in saying that in short order after this forum that we can come up with a plan, work that through the Secretary's office, and get that out to the community to react to. We're talking weeks, not months, years. Speed is our middle name. As you know, our job is to accelerate better health outcomes, and we are, as I said in the beginning, very committed to doing this quickly. To that point, one key question for me is, clearly money is required here. So I don't want to dismiss that, but what are the barriers right now? Like, why can't we be sitting in this room next year and have have that validated panel of biomarkers, or have the mechanisms of action well articulated so we can move to the next step in the game? That's the thing that we're interested in, because again, we know it's going to take money, but what are those things that are going to really give us that exponential gain?
Yeah, so obviously I do think that this can be solved with resources. I think that one of the challenges that we've had is just the timelines for reviews of proposals, to kind of get them from proposal stage to actually in action. It often takes a year or longer to get through peer review. These processes are meant to be deliberate and thoughtful, but that timeline doesn't really work for what we're talking about. And so, what I'd like to see are things like dedicated review panels with expertise in these conditions, so that we're not kind of starting from scratch explaining to someone in a different field why they should care about long COVID or these related conditions. And then I'd like to see an accelerated sort of timeline for grants in these agencies. programs like that have existed historically for other conditions that are a priority like HIV, but 18 months from proposal stage to funding at best is not really the speed that we need to be going at. Those are structural problems that I think could be addressed.
And just to add to that, because it's such a great point, is it dissuaded us from applying for grants 2 years ago. We figured that we could raise the money privately quicker than we would write a grant, have it reviewed. The 18 months, I think, is very accurate. I realized that when we were submitting RO1s back in the day. And that's an impediment. That to us, that was like, we're going to go at it alone and try and raise this money. And that, that became a slog. So, you know what? I think that is a critical point to really expedite the review process and get the money moving.
So CEDAR at FDA has a biomarker validation process program. And obviously, this is a priority for this administration. So when you have biomarkers that are well characterized, let's
say 2 years ago, and it was sent back to us. So I hope that's true, and I would love to resubmit it.
I'll follow up on that. Thank you for bringing it to our attention.
Well, we've come to the end of our session. I want to thank this extraordinary pantheon of great researchers for being here and for your commitment to solving this problem for our country. We want to work with you, and I hope you will continue to communicate to us about what we should be doing better to make this cure come faster. Thank you all very much.
Great. That was great. Thank you. That was great.